Good morning, everyone. Welcome to Biomedicum Seminar Series. I am your host, the Research Assistant at Kathmandu Research Institute of Biological Science, also known as KRIBS. Today we have Dr. as our guest. He recently completed his PhD in microbiology from University of USA and is pursuing postdoc research at and Women's Hospital, Harvard Medical School. He has published various impactful papers in various journals. The presentation will be of 20 minutes, followed with 10 minutes Q&A session. This seminar is live streamed at KRIBS Facebook page and is recorded for. Some of the housekeeping rules of the seminar are stay muted until the end of presentation, and you can ask question at the Q&A session by unmuting in the chat box. Once again, welcome Dr. Pratik. The floor is yours. You can start your presentation.
Thank you very much. Let me share my screen first. Screen sharing is disabled right now. Okay. Can you see this?
Yeah, it's visible.
Okay. Let me start. Hello, everyone. I'm Pratik Koirala, and I just completed my PhD from the University of Kansas in microbiology. And today I'm going to be presenting you with some of the research I did during that time. And the title of my talk today is Listening in on Your Neighbors. Eavesdropping promotes competitiveness in polymicrobial communities. So the first thing we need to talk about is we need to know is that bacteria exist in mixed communities. So everywhere in the world, almost everywhere in the world, bacteria exist in a mixed community. That means there are more than one bacteria present almost everywhere on Earth. And that is true for our gut, for soil and water, and even on the surface of our cell phones. And bacteria need to compete with one another for a resource like nutrients and just the space to grow. So if we take a bacteria like Chromobacterium subtsuga as an example, it needs to compete with gram-positive cocci and gram-negative bacteria, some fungi, yeast, and even viruses for nutrients and space. So it's very beneficial for the bacteria to form a group. So bacteria benefit from being part of a group. So if there are a lot of bacteria working together, then they can compete better and acquire more nutrients, more space to grow, so they can outcompete with. Oh, the screen sharing went away. I cannot screen share anymore again.
Hold on, we're fixing the problem.
Okay.
Try sharing.
Let's see. Yeah. Okay, let me continue. So bacteria benefit from being part of a group. So that way they can coordinate group behaviors and be more efficient in competing with other bacteria. And one of the ways by which bacteria compete is through this process called quorum sensing. So what happens here is when there's a low cell density condition, there's not enough cells, and they produce these signals, which I've shown here in red circles. And as soon as the signals are produced, they just diffuse outwards and away from all of these cells. But when there's a lot of cells present, they produce these signals, and these signals can diffuse back into the cell. And when that happens, all of these bacteria will start a group behavior. Like in this example right here, the cells are yellow in color at the start, but when there's a lot of cells present, they turn purple, which is because of a production of pigment. So this is how quorum sensing system works in plants, in insects, in gram-positive and negative bacteria. But the focus of my talk today is quorum sensing in proteobacteria, which is one of the phylum of bacteria. What happens in those bacteria is there are two different proteins, LuxI type protein and LuxR type protein, which are synthesized through the LuxI and LuxR genes. The LuxI is a signal synthase which makes these signals that are shown in red, and LuxR are the signal receptors. But at a low cell density condition, the LuxR proteins are almost instantly degraded. But when the cells reach high enough cell density, there's a lot of signals being produced by LuxI, and they diffuse back into the cell and bind to the LuxR protein. So now they're not degraded, but they go on to control the expression of hundreds of genes in its regulon. These little signals right here can vary between different bacteria in that they're all acyl homoserine lactone molecules, which means they have this lactone ring and the acyl side chain, but they vary in the length of carbon acyl side chain. So this one has 6 carbon in this tail, this one has 8 carbon in its tail, and they also vary with the functional group in the third carbon. So the first and the third one does not have any hydroxyl group, but the second one has a hydroxy group in the third carbon. So a lot of bacteria do this, but the signals they produce can vary. So quorum sensing controls a lot of different behaviors like production of antibiotics, which helps it kill its competitors in an environment. It controls the production of protease, which means the bacteria can degrade proteins in the environment and take up amino acids as nutrient source. It helps in formation of biofilms. That means it can take up space in the environment and proliferate much more than its competitors. And it can also produce efflux pump gene expression, which helps it survive against antibiotics produced by its competitors. So quorum sensing has a lot of different functions in helping the cells survive during competition. So what happens basically is if the cell is at a high cell density condition, they are producing antibiotics, they are producing protease, forming biofilms, producing efflux pump gene expression. And when a new cell arrives in the same environment, the antibiotics produced by these cells will simply kill the other cells, and the protease and biofilms will help it acquire nutrients easily. So the other new cells cannot compete in the same environment and they just die off. And the cells that are at a high cell density condition can grow more. So that's how quorum sensing helps during competition. So the bacteria I study is called Chromobacterium subtsugae. It is a beta proteobacteria, so it also has a quorum sensing system. It is non-pathogenic, so it's really easy to study because we won't get any diseases from it. But it is related to an environmental opportunistic pathogen called Chromobacterium violaceum. But the main reason we study Chromobacterium subtsugae is because it has a single quorum sensing system, so it's really easy to delete one or both of these genes to see how it affects the quorum sensing system. Where CVII right here is the signal synthase, which produces the signal called C6 homoserine lactone. That means the side chain has 6 carbons with no functional group in the third carbon. And another thing that's really good about Chromobacterium subtsugae is that it produces a pigment called violacin when it reaches a high enough cell density condition. So at low cell density condition, the culture fluid looks like this, like normal colored, but when the quorum sensing system is activated due to high cell density, the cell starts producing violacin, which is purple in color. So we see that the media starts turning purple. So that's how we can easily tell when there's quorum sensing system, which is activated in Chromobacterium subsegay. So to study competition in our lab, what we do is we use this thing called a dual species model, where we mix two different bacteria into a flask at an initial ratio of 1:10 and let them fight one another for 24 hours. And then we culture them again to see if the ratio changed. So if Chromobacterium wins in a fight, the 1 should go up, so the ratio should increase. And if Burkholderia wins in a fight, then this 10 should go up, so the ratio will decrease. So that's how we know which one of these two bacteria win in a competition when we culture them together. So what actually happens is Burkholderia, which is the cell right here, has 3 different quorum sensing system. It produces its own signal and uses all of this quorum sensing system to produce an antibiotic called bactobolin, and it uses bactobolin to kill this other bacteria, Chromobacterium subsegay. And this bacteria, C. subsegay, also uses quorum sensing system to produce an antibiotic to kill B. thailandensis, but we don't know what that actually is. So this is what happens in a co-culture. Both of these bacteria will produce antibiotic and try to kill each other. So, but in our experiment, what we do is we delete bactobolin genes from Burkholderia thailandensis. So this only becomes a punching bag for Chromobacterium subsegay. So it cannot attack Chromobacterium subsegay, but can just get hit by the antibiotic produced by C. subsegay. And we study how well C. subsegay can basically attack Burkholderia in a co-culture model. So this is the result of a typical co-culture experiment. In the y-axis is log of Bt over Cv. So Bt is Burkholderia and Cv is Chromobacterium. So as I just told you, the initial ratio was 10:1. So the log of 10:1 is 1. So the initial ratio is here. So as these dots go down, that means Chromobacterium wins. And if the dots go up, that means Burkholderia wins in a fight. And we see right here, when none of these bacteria can produce its own signal, the competition is almost the same as 1 right here. It does not go very far down. But when we delete AHL producing CVII from Chromobacterium, but let Burkholderia produce its own AHL, Chromobacterium is winning. Why is that? Burkholderia should have a competitive advantage in this case, but we actually see that Chromobacterium is the one that's winning in a fight. And we see that in the third bar as well, because when CV is not producing its AHL and Burkholderia is also not producing AHL, which is basically the signal. But when we add exogenous signal, chemically synthesized signal, we see that Chromobacterium is again winning in a co-culture because the dots are all the way down here compared to the initial value of 1. And when we delete CVIR, which is the signal receptor, the competitiveness goes back up again. So this tells us that even in the absence of its own quorum sensing signal synthase protein, Chromobacterium can somehow use signals from Burkholderia and compete against Burkholderia. So basically what's happening is, even if it cannot produce its own signal, it can listen in on the signal produced by its neighbor and use that information to produce antibiotic and kill Burkholderia. So to test that further, what we did was we used 2 signal synthase knockout strain of both Burkholderia and Chromobacterium and treated them with synthetic signals, all C6, C8, 3-hydroxy-C8, and 3-hydroxy-C10, because these 3 are basically signals produced by Burkholderia, whereas this one right here is the native signal produced by Chromobacterium. And we see that with C6, the Chromobacterium is very good in competition. But with C8 and C3-hydroxy-C8, it is a little worse at competition, but it can still compete when compared to the bar right here, where there's no signal at all. And there's no significance in the increase in competitiveness with 3-hydroxy-C10. So this tells us that Chromobacterium can use the C8 and 3-hydroxy-C8 signals made by Burkholderia and use that to gain competitive advantage against Burkholderia itself. So what is the antibiotic that is important for C subjugate to compete with Burkholderia thailandensis? Like I showed you in this previous figure right here, we know Burkholderia uses bactovolin, but we don't know what Chromobacterium subjugate uses. So to test that, what we did was we used RNA-seq. So in this experiment, what we do is we take RNA from 3 different samples. In absence of signal, when the cells are grown in absence of signal, when the cells are grown with C6 homoserine lactone, the signal that Chromobacterium produces on its own, and C8 homoserine lactone, which is the signal Chromobacterium can listen in on, but cannot produce on its own. So this is the signal produced by its competitors. And when we grow them in these signals and isolate total RNA and convert them to cDNA and then sequence them, we can tell which genes are being transcribed in these 3 flasks. So when we analyze the transcriptome, what we figured out was there are 97 genes in the Chromobacterium genome which are upregulated by both C6 and C8 homoserine lactone. And we know that the gene we're looking for is upregulated by both C6 and C8. That means the gene we're looking for is one of these 97 genes. And what we found was 3 of the 97 genes formed the HCN ABC gene cluster, which is responsible for synthesizing hydrogen cyanide. So that was the best candidate for the gene, the antimicrobial compound that we think is responsible for the competitiveness of Chromobacterium subjugate. And we tested that further by looking at the cyanide production in 3 different strains of Chromobacterium. So the wild type Chromobacterium strain can, if we think that wild type can produce 100% cyanide, when we delete the quorum sensing system, the concentration of cyanide went down. And when we delete the hydrogen cyanide synthase gene HCNB, and the cyanide concentration went down. So this tells us that production of hydrogen cyanide is induced by quorum sensing and also the HCN ABC gene cluster. So next, we wanted to see the competitiveness of C subjugate with Burkholderia thailandensis in all of these mutant and wild type strains. So the wild type C subjugate can compete against Burkholderia thailandensis at a normal rate. And when we delete the CVII signal synthase knockout signal synthase gene, the competitiveness does not go away. This is because C subjugate can still listen in on the signal produced by Burkholderia thailandensis, even if it cannot make its own signal. And when we delete CVIR, the competitiveness goes down because even if it can listen in on Burkholderia thailandensis, if we just cut out its ear, there's no point in listening in. It cannot listen in on anything that Burkholderia is doing because CVIR is the receptor that actually binds to the signal. And when we delete hydrogen cyanide synthase HCNB, the competitiveness goes down. And when we delete HCNB with CVII, the competitiveness stays at the same level. And when we compare this bar with the CVII knockout bar, we can see that there's a decrease, which means cyanide is important for C subjugate to compete with Burkholderia thailandensis. And we just tested the sensitivity of HCNA promoter to different AHLs by making the HCNA LAC-Z fusion. So what happens here is that if the promoter is active in presence of any of these signals, the LAC-Z gene is expressed, and we can measure LAC-Z activity using beta-galactosidase assay. So that's just a reporter assay that you can do to test the effectiveness of a promoter. And we see that the native signal C6 has the best response. The C8 is the non-native signal produced by Burkholderia thailandensis, has a slightly worse response than C6, but still a lot better than the other two signals, 3-hydroxy-C8 and 3-hydroxy-C10. So this shows us that the promoter for HCN-A gene is extremely sensitive to the cognate signal C6 that Chromobacterium produces on its own, but it's still sensing C8, but the other two not so much. So from this experiment, we know Chromobacterium can sense C8 pretty well. So what's happening right here is when C subjugate comes in contact with a huge cell density of Burkholderia thailandensis, it can just listen in on the signal that Burkholderia is making and use that for its own advantage, like start producing violacin, which is the purple pigment right here, and then use the signals produced by its competitors to produce hydrogen cyanide and just start killing its competitors. So, and then start replicating and overcome the competition right here and completely destroy Burkholderia thailandensis in a competition. But what's even more interesting than that is the set of 53 genes right here that are upregulated in presence of C8, which is a non-cognate signal, but not in presence of C6. So these 53 genes are upregulated only when Chromobacterium senses that there's other bacteria present in the environment. And of these 53 genes, a lot of them were responsible for producing type 4 secretion system, type 4 pili, some genes for antibiotic resistance, some amino acid metabolism gene, some genes to make the cytochrome C oxidase complex, and much more, which are not written down right here. But these 53 genes are really interesting because the bacteria is actively searching for the signal produced by its competitors and using that information to upregulate all of these genes. And this kind of activity of a quorum sensing system had never been seen before. So we were really interested in what these 53 genes were and how they were helping Chromobacterium subjugae in a competition. And this is just a droplet digital PCR to verify the result we got from RNA-seq. And we see that C8, which is the non-cognate signal, is upregulating the expression of these three genes right here. But C6, which is the cognate signal, is not doing that. So this pretty much confirms our RNA-seq data. And the other really interesting thing is that of these 53 genes I told you about right here, 23 of them are in a single gene cluster. And the first half of this gene cluster codes for a type 4 secretion system, and the second half codes for a type 4 pili. The type 4 secretion system is used for conjugation, DNA uptake, and effector translocation. So basically, a bacteria can use type 4 secretion system to transfer its own DNA to some other bacteria, or even uptake DNA present in the environment, and also effector translocation, which is just sending in proteins into the cytoplasm of other bacteria. And the type 4 pili is really important for adhesion, for biofilm formation, and also for twitching motility. So we wanted to test if the non-cognate signal C8 does in fact upregulate the expression of these two gene clusters by looking at a functional assay for type 4 pili. So what we did right here is we made three plates with C6, C8, and no signal. And we inoculated the bacteria in the middle and tested to see how far the bacteria can move from the center where we inoculated them. And we see that in presence of C8HSL, there's the largest motility circle right here when compared to no signal, and even in presence of C6HSL. And we can see that right here. And the area of the circle that's being produced right here is the highest in C8 SSL when compared to C6 or no signal. So what we think is happening right here is when C subjugae comes across this preformed colony of Burkholderia thailandensis or any other bacteria, it will just take up the signals made by this other bacteria and use that information to like produce type 4 pili and run away, just run away from there because it cannot compete with this huge colony of Burkholderia thailandensis. And when it's making type 4 secretion system, what it can do is it can just take up the signals from Burkholderia thailandensis, produce hydrogen cyanide, kill its competitors, and when it's dead, it will release the DNA into the environment, which C subjugae can take up using the type 4 secretion system and incorporate it into its own genome or keep it as a plasmid inside its cytoplasm and gain new genes that were previously inside its competitors. So if the competitor was making antibiotics, maybe C subjugae can now gain the genes responsible for producing that antibiotics and proliferate and grow in the environment where there was a huge competitor. So in conclusion, the research I did shows that C subjugae is a great model to study eavesdropping, such that it can produce its own signal and do a lot of things, but when there's not enough signal, it can eavesdrop on the signal produced by its competitors and use that information to produce antibiotic antimicrobial compound like hydrogen cyanide. And on top of that, we also found a novel quorum sensing mediated mechanism to detect the presence of its competitors and design a personalized attack against them and also run away from them. So it can run away when there's its competitors present, or it can kill the competitor and take up their DNA and gain a competitive advantage in that way. So with that, I'd like to end my talk right here. This acknowledging the lab that I finished my PhD from and all of the funding source that was really important to do the work I did right here. And thank you. If you have any questions, feel free to ask me right now.
Thank you so much, Dr. Pratik for this amazing presentation and shedding light between the communication between two bacteria. If anybody have any questions, you can write down in the chat box or unmute yourself to ask.
Hello Pratik Dai.
Hello, Rises. के छ?
ठिकै छ. मैले फर्स्ट पार्ट चाहिँ मिस गरेँ. मिस द फर्स्ट पार्ट. तर त्यो के भन्छ पछाडिको पार्टमा चाहिँ त्यो बर्कहोल्डेरियाले चाहिँ अनि केही पनि गर्दैन. लाइक इट डजन्ट ह्याभ एनी डिफेन्स मेकानिजम्स अगेन्स्ट एनी अफ द attacks, hydrogen cyanide attacks that the C subjugae does.
No, because so the way by which hydrogen cyanide works is it goes on and binds to the cytochrome C oxidase, which is part of the oxygen transport mechanism in the bacteria. And what happens is hydrogen cyanide goes and binds to the active site of cytochrome C oxidase, which means that the oxygen is not transported through the cell and it accumulates, produces reactive oxygen species and kills the cell. So that's common for all organisms, from us humans to the bacteria. None of us can be resistant against hydrogen cyanide, but Promobacterium itself, what it has is a mutation in a cytochrome C oxidase complex, which prevents hydrogen cyanide from binding to the active site where oxygen is supposed to bind. So it itself is not killed by hydrogen cyanide, but there's very small amount of organisms that can actually resist hydrogen cyanide attack.
I see. So is this mechanism common for other chromobacteria as well?
Yes. So Chromobacterium are known to eavesdrop on its competitors. So all the other bacteria, not all, but most of the other bacteria can only detect its own signal, and anything else will just not bind to the active site of the protein that's responsible for doing all of this. But Chromobacterium can bind to 18 different kinds of signals, and almost all of the Chromobacterium do that. So there's like more than 30 different species of Chromobacterium, and as far as I know, more than 20 of them do that, but I'm not sure if all 30 of them do this. But yes, it's pretty common in Chromobacterium and a few other bacteria, but it's not done by any other bacteria.
I think my last question is, I don't know if you mentioned this earlier, but are there any medical or biological or ecological impacts of this interaction?
Yes, so this is much more focused on ecology of bacteria than taking it into a medical field. I had two projects. One of them was taking it into a medical field where the bacteria can produce efflux pumps to prevent itself from being killed by antibiotics. And that's controlled by quorum sensing. So if we can just attack the quorum sensing system by using quorum sensing inhibitors, then that would be a really good way to kill the cells by doing a dual dose, like first inhibit the quorum sensing system and then hit them with antibiotics. That way they cannot efflux the antibiotics out. But the research I presented today is more of a bacterial ecology question where we study how bacteria are competing against one another, because that's where most of the antibiotics come from. It's not us that's driving most of the evolution in bacteria, but it's competition between two bacteria in their own ecological niche. So that's why it's important to study stuff like this. And we found a novel method by which bacteria are detecting other bacteria and personalizing an attack against them. So that could be a really nice avenue to take to study how bacteria can evolve to be resistant to antibiotics and use quorum sensing inhibitors to prevent resistant bacteria, basically.
I see. Thank you so much for sharing your study, and it was a great presentation.
Thank you, and thank you for the questions.
I have a question.
Mm-hmm.
Experiment where you remove the signal from Chromobacterium, right? And you present it with another bacteria. So at that point, that the other bacteria didn't get any signal and couldn't attack Chromobacterium, right?
No, it could produce its own signal, but it could not produce the antibiotic that it uses to attack Chromobacterium. That's just to make the experiment easier, because if Burkholderia was also attacking Chromobacterium, then interpreting the numbers would be really hard. It does produce signal, but it does not produce antibiotics. So Chromobacterium is safe, and we can just look at how Chromobacterium attacks Burkholderia without thinking about how it's defending as well at the same time.
So my question would be, wouldn't it be a safe bet to not produce signal and be at a safer side to go in a combat when competitive, when they compete?
Yes. So the environment, there's like 1,000 different bacteria, and that's in a lower number. If you pick up a glass of water from a river, there will be thousands of different organisms, and Chromobacterium is one of them, which can eavesdrop. So Burkholderia still needs to compete against 999 other species of bacteria in there, and quorum sensing system is a really good way to do that. So it would not be very beneficial for Burkholderia to get rid of its quorum sensing system, because it still needs to compete against those 999 other species. But Chromobacterium figured out a way to just exploit the quorum sensing system of Burkholderia and win against it in a competition, even when it's in a low cell density condition. So it's better to just win against 999 others than to stop the quorum sensing system. Sorry, the quorum sensing system to win against just one.
Okay, thank you so much for that answer.
Yeah.
Do we have any more questions? I don't think we have any more questions left. So anyway, thank you so much, Dr. Pratik again for this
Thank you
interaction
for
between
inviting.
bacteria.
Yeah.
We're at the end of this session. So once again, thank you so much. And bye-bye.
Thank you. Bye.